Hi there. We were discussing about the membrane biochemistry or cell biochemistry. So in this lecture, we will be discussing the fluid mosaic model, which is from the Harper's biochemistry. So what is fluid mosaic model? According to this model, membrane or cellular membranes are lipid bilayer. They are in which lipid bilayer in which proteins are embedded, in which proteins are embedded in a mosaic like pattern. So this is a lipid bilayer here, this is a lipid bilayer, and in this lipid bilayer proteins are embedded. For example, these proteins are embedded here, these proteins are embedded here, and these proteins are embedded here, and these proteins can cross this membrane as an integral protein as well. So this is a basic concept of fluid mosaic model of the cellular membranes, which states membranes are lipid bilayer having protein embedded in them in the form of in a mosaic pattern. However, this membrane shows phase changes. According to fluid mosaic model, this membrane shows phase changes. It changes its phase from fluid to solid or vice versa, or you can say fluid means increased fluidity and solid state means decreased fluidity, and increased fluidity also increases permeability and decreased fluidity shows decreased permeability as well. So this membrane shows phase changes from increased fluidness or increased fluidity to decreased fluidity. Increased fluidity also increases the permeability, while decreased fluidity decreases the permeability, and the temperature at which its state changes is termed as transition temperature. Transition temperature at which its fluidity decreases. Okay, so how can we explain this? How can we explain this? Uh, let me explain. The fluidity of membrane also depends upon the structure of the structural lipids. It depends upon the structural lipids of the membrane. For example, we previously discussed that if lipids are saturated, they have straight tails. Saturated lipids have straight tails. However, unsaturated lipids have tilted tails. They have tilts in their tails. They have tilted tails. Okay. So another concept is we know that in solid state molecules of substance are closely packed or densely packed. This is solid state. However, in liquid state they have gaps between them. Okay. However, in gaseous state these gaps are much more. So this is solid state, this is liquid state, and this is gaseous state. Same concept we will be applying in the lipid membranes, lipid bilayers. So if the membrane lipids are saturated, they will be tightly packed. Saturated lipids will be tightly packed in this way. Okay. However, if the membrane lipids are unsaturated, if they have tilted tails, these tilted tails will need more space. They will need more space, so lipids will be pushed away. So supposedly this is a membrane here with unsaturated lipids. So in this case there will be tilt in membrane. This tilt in membrane will be needing more, you can say, space. So as a result these lipids will be less tightly packed. Okay, so these lipids are less tightly packed, or you can call it disordered, and these are ordered. So they will be having less fluidity, while they will be having more fluidity. So unsaturated membranes having unsaturated lipids have more fluidity, while saturated one have less fluidity, because saturated are densely or more orderly packed. Moreover, if we increase the temperature of this, temperature here is increased, what will happen? We know that temperature increases the energy of molecules and molecules begin to vibrate more violently. So what will happen? Increasing temperature will push these lipids far away from each other. So what will happen? These lipids will be arranged in this way and there will be some space between them, and this here it will be space like this, and temperature will increase the fluidity of these ordered lipid bilayers. So they will be changed from ordered to disordered state, and it will increase the fluidity, and the temperature at which the fluidity will increase, or they will attain from solid to, you can call it a liquid configuration or fluid configuration, is called transition temperature. Okay, transition temperature, or it is a melting temperature of the cellular membranes. So moreover, it is concerned with the permeability of membrane. When fluidity increases, spaces are created. These spaces allow the passage of materials. However, in this state, the space for passage of material is less. However, when they have moved away, space for passage of material has increased. So increased fluidity increases the, when fluidity increases, it increases the permeability of lipid bilayer. Okay. So it was a basic concept. However, the substances which move by channels, for example, here is the channels. This effect of membrane membrane fluidity on the channel is less. Okay, so channels effect of membrane permeability on channel protein is less. However, the substances that directly cross the lipid bilayer effect is much more. Okay, so another concept here is the role of cholesterol or sterols. Cholesterol is a large molecule. Cholesterol is a large molecule and it plays a buffering role. It plays a buffering role in the fluidity of cellular membranes. How? If when the temperature is less than Tm, it should be membrane should be in solid or less fluid state. However, these are supposedly lipids of the membrane. So cholesterol is a large molecule. It interrupts the tight packing of the lipids below the Tm or below the melting temperature. So in this way, it somehow prevents it from getting too much hard. So it keeps some fluid nature of the membrane. So when temperature is less, ideally membrane should be in the much tightly packed configuration. However, what does the cholesterol do? Due to its large molecular structure, it interrupts lipid from getting too much close and maintains some fluid nature. However, when temperature increases, again due to its large structure, it prevents the lipids from getting too much away. It prevents from getting too much lipids. It prevents lipids from getting too much away. So again restricts the fluidity. So cholesterol plays a buffering role and keeps the fluidity of membrane in a specific range. So it was a basic overview of the fluid mosaic model, and it is again we discuss it from the Harper's chapter 40 of the Harper's Biochemistry, the fluid mosaic model. Here we discuss the basic concept of this from the Harper's Biochemistry. Thank you.